Good morning, everyone on campus, and good morning, good afternoon, good evening, everyone on the Zoom. And I'm a group leader, full DCP group leader here at Janelia. And today we are going to start today's science with proteomes and metabolomes. And our first speaker will be Marian, who is going to talk about genome-scale models of metabolic wiring. Thank you. And rewiring.
Okay, can everybody hear me okay? Great, thank you. Thank you so much for the organizers for having me here today and share our work. So we're going to be talking about metabolism in this first talk. Turn on thing, node to self. Okay, here we go. So we all know that metabolism is important for everything every cell in your body does. What metabolism is the conversion of dietary nutrients into biomass and energy during development, in wound healing, and for daily processes. Now, our metabolism is governed by a complex network of metabolic conversion reactions, as illustrated here. And so it's through these networks that are different. Well, the network is the same in every cell, but it's wired differently in different cells, that cells do what they have to do. And so I'm going to be talking about wiring and rewiring today. And so first I would like to tell you what I mean by that. So here's the metabolic network of an organism. And so in each of the cells in your body and under different conditions, different parts of this network carry flux or are used to do what the cell has to do. And so the analogy has been drawn by Rabinowitz's lab with traffic. So the network itself is like Google Maps. It doesn't change. The roads are always the same. However, which parts of the network is used is called wiring. And so the cars are analogous to metabolites that flow through the network through conversion reactions. And the rate of individual reactions, so the conversion rate per time, is called flux. And this is akin to flow of traffic through the roads. Okay, when this flow is perturbed, same as in traffic, metabolism can decide to use another route. And so this is what we call rewiring of metabolism. We know that rewiring of metabolism is important. Of course, everybody knows about metabolic rewiring in cancer, where cells change from using glucose as a carbon source to glycolysis to help make all the components the cell needs to grow rapidly. All right, today I'll tell you about our work in the nematode C. elegans, in which we ask how is metabolism wired in this organism, and also how is metabolism rewired in this organism to maintain homeostasis. And this can be under different perturbations that are genetic or diets or diseases and so forth. And so if you want to understand flux of a metabolic network in detail, you would have to measure flux of every single reaction in the network. Our network has about 2,000 reactions, and so you would have to do isotope tracing with conventional methods to determine flux rate of each reaction, pathway, and then the network. And this is incredibly hard. And so if you want to do this at the network level, you would have to measure every single metabolic reaction. And if you want to understand rewiring, you would have to perturb every metabolic reaction followed by isotope tracing. And this is too hard to do right now. So what I hope to convince you of today is that we have changed this problem of metabolomics into a genomics problem. And so what we did was we thought, well, metabolism is often regulated at the level of gene expression and of course also by allostery, which we won't talk about. There's several famous examples of the control of metabolism by transcription, all the way from the lac operon in bacteria, the GAL4 system in yeast, SREBP in mammals, and we discovered that conditions of low dietary vitamin B12 result in a transcriptional rewiring of metabolism in the nematode C. elegans. So there's two reactions that use vitamin B12 both in you guys and in worms. The first one is here in the breakdown of the short-chain fatty acid propionyl-CoA. And what we discovered is that the animal, when it gets a diet low in vitamin B12 or when it has genetic perturbations in this pathway, transcriptionally activates a set of genes that constitute an alternate pathway, thereby rerouting metabolism in sort of this beta oxidation-like breakdown of propionyl-CoA. The other vitamin B12 using pathway is the MET-SEM cycle, which generates acetylmethionine, which is the major methyl donor of the cell, in which methionine synthase uses vitamin B12. In this case, when B12 is low, the animal upregulates enzymes within the pathway to try to compensate for low flux through the cycle. And so we really want to know is, in this animal, how metabolism is wired and how it's rewired. And what we aim to do, and I'll show you, is we want to measure every metabolic gene and we want to perturb every metabolic gene to convert this metabolism problem into a genomics problem. If we want to do that, of course, we first have to know what C. elegans metabolism is like. And so a couple of years ago, 9 years ago, Shafak Yilmaz, who's a bioengineer in my lab, reconstituted the C. elegans metabolic network, and we did some tissue analysis then so that we know what this map looks like, this Google map looks like for the animal. And then in 2 studies that I don't have time to talk about, we found that C. elegans metabolic genes are extensively transcriptionally regulated in a computational study by Shivani Nanda when she was a graduate student in the lab. And we also know that enzyme levels, by and large, are predictive of metabolic flux. Other people have studied this as well, and Zhuhang Li has recently published a paper in which we expand on this at the pathway level. Okay. So now, we'll go back to the question, and I'll show you how we do this. So last night we heard a beautiful talk from Aviv Regev, and I don't have to explain what perturb-seq is, pioneered by Jonathan Weissman here in the room and Aviv. What Zhuhang Li and Hefei Zhang in my lab did is they developed a method that we call worm perturb-seq. There are some key differences with perturb-seq, but it's inspired, of course, conceptually by the perturb-seq studies. So in our work, Fei and Hang systematically perform RNAi of metabolic genes in the whole animal, followed by whole animal RNA-seq that combines, it's all technical stuff, that combines advantages of bulk RNA-seq and single-cell RNA-seq in high throughput. And then we use a novel statistical framework to identify differentially expressed genes. Very, very briefly, I know that Aviv said yesterday, if you have a lot of quantity, you don't need so much quality, but we still like to have really good quality differentially expressed gene calls. And so what we found in our studies is that most genes, the distribution of its expression many genes doesn't quite follow the theoretical null model. Some genes are just noisier, as indicated by this red distribution. And so what we did then is to use the entire data set that I'll tell you about in a second in a gene-centered way, to find out those conditions where each gene is differentially expressed. So we have these null distributions for every single of the, say, 10,000 genes that we find detected by RNA-seq in the animal. And so the differentially expressed gene calls that we make are of very high quality. Okay, so Fei and Hang did this worm perturb-seq for 900 metabolic genes out of the 1,314 that we have in our network in triplicate, 6 million reads on average, and then derived a metabolic gene regulatory network that connects the 380 or so responsive perturbations where we do find differentially expressed genes. Happy to elaborate on that later. Connecting to about 9,000 differentially expressed genes through 110,000 regulatory interactions. So now we can start answering those questions on how metabolism is rewired in this animal when you perturb its own metabolism. And so what we find is that, same like the MET-SEM cycle, we find about 40% of perturbations, we find some intra-pathway compensation. We also find rerouting that's harder to quantify, actually, because pathways are all connected, but we have several clear examples. And then we also find these very complex coordinated subnetworks in the whole network that seem to be coordinately responsive when perturbed. However, these 3 modes explain only about 10% of all the interactions among metabolic perturbations and metabolically differentially expressed metabolic genes. And so to gain more insight into what's going on at this level, we did exactly what Aviv also talked about last night. We looked at the structure in the data to identify perturbations that are similar. So perturbation A, perturbation B give the same differentially expressed genes on the one hand. And on the other hand, differentially expressed genes that respond the same to sets of perturbations. And so now we can connect 22 perturbation clusters to 44 what we call gene expression programs, exactly what Aviv talked about yesterday, and to see if there's any patterns there. So that's what you see here. Of course, you can't read this in any detail, but you see that there's structure in this data. And so then when Fei and Hang manually started to look at this, we saw a very simple principle that started to emerge. Here, genes are functionally colored, so this is energy, fat synthesis, and so forth. And we see that when you perturb energy, for instance, energy genes tend to go up. So Fei and Hang manually looked through this data and devised this network of sort of what we now call core metabolic functions and how they respond to different perturbations. Now, this is all manually annotated. We say, oh, this gene is involved in lipid synthesis, and oh, this gene is involved in energy generation. And so, of course, that's not very rigorous. So we took advantage of our metabolic network and tools to model the network that are called flux balance analysis. And I'll tell you how that works right now. Okay, so for every gene and therefore reaction in the network, we can computationally ask, when you perturb this reaction, for instance, does it have an effect on energy, fat synthesis, and so forth, using flux balance analysis? Again, I won't go into the details, but it's statistically rigorous. So we have these 5 core metabolic functions: energy, and then the 4 major parts of the animal's biomass: protein, lipid, extracellular matrix, and nucleic acid. And we see this is, you don't have to look at the details, this is the assignment of the genes in the network to these different core metabolic functions. That is now not manually, obviously. And then we can ask, this pattern that Fei and Hang saw, does this hold true when you do it in this rigorous way? And so the answer is yes. And so what you see here is every row is a perturbation, and then these colors indicate the target genes of that perturbation and their associations with functions. So this gene has this proportion of target genes involved in energy, this in fats, lipid synthesis, and so forth. And so what you can see here is that perturb an orange genes go up, perturb a green genes go up, and so forth. And so this is just for the purpose of illustrating, but we can, of course, now quantify this. And Fei and Hang named this the compensation-repression model, where genes that compensate for loss of a certain reaction by RNAi in this case, and they repress other functions, most likely to maintain homeostasis. Okay, so now we can quantify this. For instance, I'll show you here, this is RNAi of one gene, GSPD1. It's associated with lipid metabolism extracellular matrix, but not with energy, protein, or nucleic acid. And then we look at the genes that change in expression when you perturb this gene, and we find that 39 of the 58 that go up are associated with the same core metabolic functions, whereas 18 of the 27 that go down are associated with the 3 functions GSPD1 is not associated with. So that together is 39 plus 18 over the total is 67% of the differentially expressed genes are explained by this very simple model of compensation and repression. Of course, we can look at this throughout the whole data set, and we found that when we do randomizations, 10,000 randomizations, we never see this level that we see in the real data set, indicating that the p-value is at least lower than 10 to the minus 4. Okay, so that's the rewiring story. There's lots more to talk about for that, but I don't have time. I want to tell you about the second story that we just published from the same data set. Because Fei and Hang had what is, in my opinion, a magnificent insight in that they could use this data to understand how metabolism is wired in the first place without any perturbations. So far in the field, what others and ourselves have done is to just use flux balance analysis and levels of enzymes, mRNA or protein, to predict flux throughout the whole network. Now, what happens then is that for every single reaction, you get a window or a range of where flux could be. And that means that for the overall network, you have a solution space. And there are many solutions that fit what you're trying to model here. And so it's not very precise and certainly not very quantitative. And so I'll introduce to you 2 metrics from the WPS dataset, responsiveness and similarity, that help to reduce the solution space. And now we get semi-quantitative flux predictions for every single reaction in C. elegans for the animal as a whole at the stage of its lifetime where we did the experiments. Okay, hold on. All right, let me take you through these 2 metrics. The first one is ridiculously intuitive. It just posits that if you respond a reaction and you get a transcriptional response, there's probably flux through that reaction. Nobody will contest that, I think. And then we demonstrated extensively that this is true. The second metric is a little bit less intuitive and deals with branch points. For instance, say that flux goes here and it has 3 options of where the metabolite can go in the network. We found that reactions close to each other in the network tend to give similar gene expression changes when you perturb them. And so now the similarity metric posits that if perturbation of this reaction gives the same DE gene profile as this gene and this gene, then we posit that flux goes this way, and you can do this sort of semi-quantitatively. And we also demonstrate that that works well. And so Zhu Hang devised a novel algorithm that combines responsiveness, DE gene similarity, but also expression levels that we have from our own data set. Together with flux balance analysis, we have a new algorithm. And now we can predict how metabolism is wired through the entire organism. And that is shown here using genomics data. I think it's beautiful. It's not only beautiful, we can also learn things about the animal's metabolism that we had never seen before and that were completely unexpected. For instance, we find that flux goes cyclically through the pentose phosphate pathway in this animal, as predicted by the worm perturb-seq data set. In textbooks, this doesn't really happen. There's only a few papers that we could find where it's in cancer cells that are in oxidative stress or bacteria under oxidative stress. But these are happily growing worms, not under any stress, at least not intentional. And so the model predicts that flux is cyclic, and it uses bacterial RNA as a carbon source. We then went on, and I won't torture you with all those details, but we then went on to use all these different glucose heavy isotopes to demonstrate that this is indeed true, many different ways of doing that. And we also demonstrate using heavy adenosine that indeed that RNA is a major carbon source to fuel this cyclic cycle. Nice. Why we think the animal is doing this is at the young adult stage. The main goal of the animal is to make babies. Babies need a lot of fat, and to make fat, you need a lot of NADPH, and the pentose phosphate cycle, one of the things that it does is to make NADPH. In cancer and bacteria, it's thought to make NADPH as an antioxidant, but here we think that it's to make lipids for the babies. Secondly, we found that flux through glycolysis is relatively low, indicating that glucose is not a major carbon source for the animal, but rather the model predicts high flux through amino acid breakdown. So now I will show you this data where we used heavy essential amino acids, because the non-essentials you can't interpret, and asked how much of that heavy carbon ends up in citrate in the TCA cycle. And then we can add that all up and find that of amino acids, at least 30% of the animal's energy comes from amino acid breakdown. We also find that propionate, or that's what I talked about earlier, is a significant carbon source for the animal, and the propionate shunt is most frequently regulated, which was very relieving to me, not to study a niche pathway. Whereas pyruvate, which comes from glycolysis, is only 12% of the animal's energy. And when we now look actually after what E. coli, which is the diet of the worm, is made of, it's mostly made of protein and not glucose. So that actually totally makes sense, except that nobody thought about it beforehand. So we find that the pentose phosphate pathway is a cycle. RNA is a major carbon source for the animal, and energy is derived mainly from amino acids and not glucose. This can change during the lifetime. For instance, the animal doesn't break down fat because, as I just told you, it needs to build fat to make babies. So overall, I hope to have convinced you that we now have a genomics lens on a metabolism problem. We can measure every metabolic gene, we can perturb every metabolic gene, and really get systems or network level insights into how metabolism is wired in the organism and how it is rewired under different conditions. And so moving forward, we want to, of course, understand how this works. And I will need both money and people. We want to study detailed pathways and genes. We want to understand the mechanisms, meaning what metabolites are being sensed by the animal and how, which transcription factors govern those transcriptional changes in the CR model, for instance. We're doing a lot of additional worm perturb-seq right now in hope to move to mammalian cells as well. And so with that, so on time, I want to thank the people in my lab, most notably two amazing trainees. Zhuhang Li, who was a graduate student, is now briefly a postdoc in the lab, will be moving on to a faculty position soon. Same for Hefei Zhang, he was a postdoc in the lab, and this is just I'm so fortunate that these two trainees work together so well to make this work possible. And this is all with my long-term collaborator, Shafak Yilmaz, bioengineer in my lab, and other people, and several collaborators who have contributed to this work. And with that, I'm happy to take questions. Thank you.
Can you comment about cell-specific energetics? Because apparently different cells have different demands and everything. And did you integrate it with single cell data, or do you have some examples when cells are radically different?
Right. So we are using single cell gene expression data to model the animal's metabolism at the cellular level, yes. Fei and Hang, when they are independent, are also working on combining this with single cell perturb-seq, so perturbing the whole animal, but then single cell RNA-seq. And they have been working on that, but now they're looking for jobs. They've been working on that as well. So we absolutely want to integrate that moving forward. And then we are now actually in the process of finalizing yet another algorithm with which we can also impute data from single cells to layer on top of this and get more and more high resolution. Are energy demands different for different cells? Absolutely. Muscle uses much more energy than neurons, and we can see that in the gene expression data.
Wonderful talk and amazing work. I had a question about the role of prior knowledge in your workflows. In a strict sort of holdout setting, you would sort of save it for the end, right? You'd make some network out of your data, and then you'd see how many edges do we recapitulate that's known. It seems like in this workflow, they're kind of there in the beginning already. Like maybe they help you interpret the data or you use it as some prior. So I just wanted to hear more about your workflow.
Yeah, so an often misconception is that the network changes. The network doesn't change. The roads are the same. So this is in contrast to gene regulatory networks, right, which really indicate information flow or signaling networks in a cell. But what we start with is just the map of all the roads. And then the wiring is when I drive to work, on a Monday morning, I always use 190 in Massachusetts, and that's the wiring that I use on a Monday. And then if there's an accident on 190, maybe I have to take off roads to get into work, and that's what we call rewiring. So the only prior knowledge that we have is the Google Maps. That's it. And then the rest is all fueled by experimental data, mostly of worm perturb-seq here, but also of the RNA-seq data. It's actually still the same data set of the unperturbed animal as well. I hope that's clear. I'm happy to talk to you more after if you want. Should I throw this thing?
Go. So I have two questions. The first one is, did you investigate whether these principles of rewiring that you explained to us depend on the diets? And second question is, in a way, what I understand is a description of fluxes without timescales, nonetheless, because this is based on what you reported. Nonetheless, I think that having an understanding of the timescales of the fluxes, depending on functional needs, may be really important.
Totally.
Could you elaborate on how you can approach that?
Yes. So timescales definitely are important, right? Because flux itself has a time component in it. So to do that, we would have to we actually have a developmental RNA-seq data set, so we can start modeling that. There's also single-cell RNA-seq data not by us, from the embryo and from the L2 stage, and we have been using that to model it. Another part that relates to your question is we do the perturbation at the first larval stage of the animal, and then we measure the RNA at the young adult stage. And so it's already after some form of adaptation, and we're fully aware of that. And then what was your first question?
The impact of diet on the rewiring.
Yeah, impact is my favorite question. Why didn't I remember that? So the whole propionate business, we all found out by changing the diet of the worm. That's how we got into all this metabolism stuff. So diet is hugely important. So right now, Zeynep Mirza, who's a postdoc in the lab, is testing like 200 different diets, same thing, to see what happens. We know that, for instance, Komomona's diet is high in B12, and you don't need the shunt. We've also published a study with Bacillus subtilis, where the bacteria deliver entirely different fatty acids. And we can see that even though the worm development is deterministic, 959 somatic nuclei, its fat is actually different depending on which diet you give it. So that's a huge question, and we are definitely investigating that. Thank you.
Beautiful talk, amazing. In the first part about the compensation-repression, it seemed like, I mean, just looking at the data, there was a lot of green. It seemed like sort of maybe a second thing that happens is that the lipid pathways get re-regulated, especially when you did protein perturbations. Do you have any biological idea about why that might be?
So very good eye. Yes, we see lipid synthesis affected a lot. Vacuolar ATPase as well. And then there's different parts of lipid synthesis and lipid breakdown that are affected under different conditions. And we would honestly have to go look back in the data set to parse out what that means. I think what we learn at sort of a very high level is that lipids are incredibly important for the animal. And as I was just commenting on when you give them Bacillus as a diet, right? Bacillus has monomethyl branch chain fatty acids. E. coli has cyclopropane fatty acids, and Komomonas, another bacteria, has mostly monounsaturated fatty acids. And you see that those all change, but then down to the PUFAs, polyunsaturated fatty acid, that all stays the same. So it's very interesting. So that means that the animal knows or senses its level of PUFAs and then is capable of using whatever diet it has or whatever perturbation it has to maintain even homeostasis at the level of polyunsaturated fatty acids. There's a huge amount of stuff to learn just from this data that we haven't had time for yet. Yeah, you're right.
A very beautiful talk. At the beginning, before you started talking about the transcriptional control, you pointed out the allosteric control is important too. Can you return to that and sort of layer that on top? Like, how should we be thinking about the two systems and how they work together?
Because when we started out, I gave talks about transcriptional regulation of metabolism, and now I put the slide in of all the Nobel Prizes and then the little bit that we have done, right? Biochemistry departments think it's all about allostery. We don't include that now. The paper that we just published in Molecular Systems, I'm not about to say that, but there's a few Lee et al. You'll find it or you email me. We find that changes in levels of an enzyme often are predicted of flux, but you get much better predictions if you take the whole pathway into account. And now we have an algorithm in that paper that you can use even for reactions for which the enzyme doesn't change in levels, but surrounding enzymes do. So say that an enzyme is regulated by allostery, which you don't see in gene expression data. Now you can look at changes in enzymes surrounding that reaction and still predict for the allosterically regulated enzyme. Actually measuring allostery at a network level scale is, I don't think it's doable yet.
Great. A question from Zoom. Running flux balance analysis requires objective functions. If you perturb genes, the objectives of cells might change. They might want to keep biomass or ATP demand constant or change. How do you account for this? And do you change them accordingly?
Yeah, thank you for that question. So we have the normal constraints that we put on the metabolic network modeling. You cannot not make ATP, for instance. So you need to have a base level of ATP. You need to have all the biomass sort of in balance. And then we used flux balance analysis here. We devised a set of reactions that recapitulate the core metabolic function. For instance, the last step or the first step, I actually don't remember, of biosynthesis of fatty acids, you can measure the flux computationally through that reaction when you perturb reactions somewhere else. So that is what we measure. We don't change anything else on the constraints of the network. Jared.
Hey, Marian, that was great. You made the point very clearly that when you knock out energy genes go up, and fat genes go up. When you looked at what genes are downregulated, it was different, but it was not random. And I wonder if you could maybe comment on that. What genes go down when you knock out genes involved in energy production?
So what we see is that the genes that go down are the genes that the perturb gene itself is not associated with. Those are the core metabolic functions the gene itself are not associated with. That's what we observe in the data. Then what I'm about to say now is complete hand waving, right? Because so what we think is that the animal, as we don't have any dynamics in the system, constantly measures or senses its state of metabolism, and then this is up and then that is down to maintain homeostasis. And that's why I think it can adapt to many different conditions. Now, even more hand-wavy is worms are incredibly successful on Earth, right? Nematodes are everywhere. Diets are different, which we talked about, temperatures, all these perturbations. And so what I think even more so is that the animal is amazingly adaptable to all these things by pushing that down a little bit and pushing that up a little bit. So, yeah.
Final question, G2.
Yeah. So, I mean, again, fantastic talk. My question is more about this idea of homeostasis that you just alluded to at the end, where sensing, of course, for each of those perturbations are likely to occur through different pathways. And so can your data speak to any of that sensing and response mechanisms? Yeah.
So this is a main question that I, again, need people and money for to do, is I would love to know how many solutions are there? How many metabolites, either by being up or by being down, can be sensed? Through how many pathways can this then lead to transcriptional changes? For instance, in the technical paper, we also did RNAi of 100 nuclear hormone receptors, and we can then integrate those data sets and find nuclear hormone receptors talking to these different gene expression programs. But it's not very crisp yet, so we can't really answer the question of how many paths are there to a certain solution. Given the magnitude of the number of changes that we see, I would be surprised if there's one solution. Also, given the fact that C. elegans has 275 nuclear hormone receptors and we have 48, and those are often involved in sensing its endogenous metabolism, I think there are many solutions to again, for this balance and homeostasis type thing, but it remains to be studied.
Thank you, Marian, for this fantastic talk.
Thank you.